Thank you everyone for joining us to this session of the World Medical Innovation Forum. The panel here is Oncology's New Paradigm. With me today is a very exciting panel of distinguished scholars and researchers and physicians who will guide us through the path in terms of the revolutionary advancements in oncology, ranging from immunotherapy to precision guided technologies and early detection, and how all of this evolved into our modern suite of targeted therapies, and looking into the future, pioneering drug discovery and biomarker strategies, all of which to improve outcomes and well-being. So thank you everyone for joining us. During the discussion, if you have a question, there's technology here. I have an iPad, but if you flag it to me via your phones and the app, we can ask the panel specific questions. So maybe to start, because all of these people are certainly more distinguished than I am and can encapsulate their research better than I can, I'm going to ask each of the panelists to introduce themselves and describe what they think is the single greatest advancement in our ability to treat cancer, or alternatively, the largest unknown stymieing further development. So working our way from right to left. Gad?
Hi, I'm Gary Getz. I'm faculty at MGH, and I'm working on cancer genomics and computational biology, next generation sequencing, recently most interested in resistance to therapy and cell surface markers, and of course using newer technologies of single cell and sequencing. In terms of the thing that I think a big revolution is more, I would want to look into the future, and I think combination therapies is where we need to go, because each individual therapy, there are resistance mechanisms that arise, but when you give multiple drugs together, there's a possibility to really eradicate all the cancer cells, which we want to do. And that, of course, ties to early detection, because you want to do it when there are fewer cancer cells.
Good morning. I'm Shannon Stott. I am faculty at the Krantz Family Center for Cancer Research at Mass General. My background is engineering, so I focus on developing new technologies for thinking about early detection as well as prognosis for cancer patients. Specifically, I'm interested in circulating tumor cells and extracellular vesicles. But I also have an interest in biopreservation of complex fluids, so to make sure that we get most information out of those fluids that we're thinking about at the time of acquisition, that we actually have all of that rich content stored. So when I think about what has made the most difference in my particular area for how we can hopefully treat cancer patients more effectively and as early as possible, I really just think of the advent of sequencing and how much those advances over the past years have allowed us to both develop better technologies, get to things earlier, but even thinking about doing molecular readouts in the home setting when I think about the future.
Good morning, everyone. Jonathan Carlson, MGH Center for Systems Biology. I'm a chemist and a hematologist oncologist. I do a lot of work in the clinical hematology space, but scientifically, the chemistry to oncology connection is what has me here with you today. We're working on building new molecular machinery for in vivo chemistry. There's an area that's called perpendicular to biology chemistry, or bioorthogonal chemistry. And it's an exciting space with molecules that are capable of finding each other and doing chemical reactions in spite of the biological context. So you can imagine delivering therapeutics that become active in situ and turning molecules off when they've wandered where you don't want them. And so in that thematic space, lots of opportunity to build new molecular machinery, and that's my area of interest. As far as the lead-off question of most important advances, I think Russ will say more and be more specific about it, but the immune system being brought to bear against cancer in a multidisciplinary, effective way is a space where we've made huge progress in recent years, and I think there's much more to come.
So I'm Russ Jenkins, also from Mass General Hospital and the Krantz Family Center for Cancer Research, and I'm a melanoma medical oncologist 1 day a week, and the other time I run a translational cancer research lab at Mass General focused on different strategies to overcome resistance to cancer immunotherapy. And a large part of how we approach that problem is essentially saying that the best model of human cancer is human cancer, and we're using a lot of patient-derived tumor models to inform the strategies that we're hoping to develop to overcome resistance to cancer immunotherapy, and in particular immune checkpoint blockade. And I would say, again, in response to the opening question, I think cancer immunotherapy, without a doubt, has been the biggest advance. I wouldn't say exclusive to immune checkpoint blockade. Obviously, cell therapy has been absolutely instrumental in a variety of different hematologic malignancies, and there's certainly more to come. And then more recently, another form of cellular immunotherapy using a patient's own immune cells as opposed to an engineered form, using a patient's own natural immunity to try to overcome resistance. And in terms of where to go from here and the biggest challenges, I think, while I could say easily overcoming resistance to immunotherapy is the biggest challenge, I think beyond that, it's going to be personalizing that approach, figuring out a way to get the best treatment, the right treatment to the right patient at the right time.
Hi, I'm Greg Simon. I'm the token history major and cancer patient, I hope, the token one on the panel. My background, I spent a lot of time in Congress on the Science Committee and in the White House with Al Gore, and ran the Biden cancer moonshot in 2016, and started Faster Cures with Mike Milken 20 years ago in the Melanoma Research Alliance. I think the biggest breakthrough in my view, because I worked on this for 20 years, is the presence of ubiquitous and interactive medical records. Because we finally can let patients get involved with their own care and get educated about what's going on. In fact, we've now gone to the point that I get my biopsy results 2 days before my doctor calls me about them. So we have to deal with that. In terms of what I worry about, we have the ability to get very granular now with cancer detection. We'll talk more about that. I think we're going to see a lot of things beyond CTC. But the biggest problem right now is still this problem. The only technology that works to screen for cancer is one person telling another, have you been screened? Everything else will not get them into the door, which is why my runner-up for most important thing is Obamacare. Because the biggest indicator of who survives cancer is who has insurance. Without insurance, you don't get checked. And if you did get checked, you couldn't afford it. So 40 million people are now in the system, and we can do a lot to save thousands and thousands of them from cancer because they will go to the doctor.
Well, I'm Keith Flaherty and co-moderator of the session. Go to the mic.
Oh, you do know.
Stole my mic.
Here comes the mic.
Well, maybe to start things off, Ross, when we think about your work doing living patient biopsies, maybe you could talk a little bit about what the current landscape looks like, and then moving ahead, as we think about trying to pair up a patient with an individual or specific treatment, is this going to be something where not only do we have a personalized kind of regimen, but potentially a tumor-specific regimen, given the heterogeneity of the tumors?
Yeah, it's a great question. So I'd say broadly now, the sort of work that we're aiming to do, the best analogy is what happens if a patient has an infection in the hospital, or if you go to your primary care doctor and they send a culture off, and you take that culture and then you test a bunch of different antibiotics against it and find out what's the best antibiotic for a particular infection. We do personalized medicine all the time in the hospital. So in some ways, it seems like it should be straightforward, but some of the key items about heterogeneity and whatnot are challenges when we just think about taking that paradigm and applying it.
What does deep molecular analysis of cancer tell us about what we're missing in currently available model systems?
So in terms of we're missing two major things. One, we're missing non-coding parts of the genome. Most assays today, most diagnostic companies do a single time point panel of gene sequencing, where we're missing the rest of the exome and the rest of the genome with translocations, inversions, and non-coding drivers, which we actually don't know all of them yet in the research. So clearly we are missing a lot in that. And the other thing we're missing is the dynamics. We are typically taking one time point, And we're not modeling or monitoring the growth and death of different subclones within the cancer. And that could be done with deeper sequencing and wider sequencing, so whole genome sequencing across multiple time points from blood or CTCs or biopsies, and then monitoring the patient's response to the therapy. So these are the main two things that I think we're missing, and that's just on the DNA level. There's methylation, there's RNA, there's immune interaction with the cancer cells and things like that, so that's additional things that we're missing. With spatial imaging of a biopsy, we might be able to get all this interaction of the immune cells and the cancer cells, which are also something that'd be useful for deciding how to treat the patients.
Great. So, yeah, Jonathan, that's your kind of your business.
Yeah.
So maybe you take that baton.
Yeah, sure. A quick segue. Right now, our technologies often make a split between watching the system evolve when it's alive in a very limited way, or stopping it, taking a biopsy, a single sample, and ending its living sequence of events, and then doing all of the highest tech breakdown that we can of it. And so one of the areas that I think there's opportunities to change broadly our approach is to use the kinds of chemistries that I was alluding to before to bring multi-channel, multispectral, deeply informative layers of information to the living tissue. And we won't be able to get to the 3 billion base pairs of the genome or all 20,000 genes in the exome. But dozens of targets in a living sample in one of Russ's engineered model systems should be doable. And I think broadly, just as food for thought, if we start asking the question, can we do multiplexed science on a sample that's alive, learn dozens of things about it, and then watch it work, we'll learn a lot more about how cancer and the immune system interact. And we're just missing all of that right now because we can't watch those systems in their full complexity. And we really haven't built out the tools to do that yet. And so, that's where chemistry can add a new set of capabilities.
So, but Shannon, taking that set of thoughts into, modalities for trying to monitor, in living human beings, the state of cancer biology, up to and including tumor immune interactions. I mean, maybe expand a little bit on sort of what compartments, what components, if you will, of any of that do you see as being sort of shining the brightest light in the near future in terms of thinking about trying to understand these questions in people?
Great. Thanks, Keith. It's a great question. I think one of the things that our laboratory is particularly interested in is not only looking at the tumor components that are in circulation, whether it's ctDNA, circulating tumor cells, or extracellular vesicles, but also pulling out messages that are being released from the immune system at the same time, because you have that first snapshot in the tumor, but you have lots of other environmental factors and other things that are occurring with this patient in terms of their treatment course as well, and being able to dynamically monitor that in real time is also going to be advantageous for monitoring their treatment, looking for toxicity to the particular treatment that they're in. So one of the things that we're doing is looking at the extracellular vesicles that are released from T cells as well as B cells and other innate immune cells that we can find then in a sample of blood. So I think it's that combination, but as Gady said, also watching and doing that. So we've been doing that with our microfluidics, but also expanding beyond just thinking about blood, also looking at saliva as well as urine that's specific to the particular cancer that we're hoping to monitor.
Maybe I'll sneak one more question before letting Jason get a word in edgewise. So Greg, much of the through line between these investigators and the technologies that they've been trying to sort of translate or apply is that if we could find cancers at a much earlier point, I mean before our current screening methods allow, or in patients who've had a cancer diagnosis, so using conventional diagnostic strategies, where we can find trace amounts, if you will, of residual cancer, right, and have move from what we've called for decades and decades so-called adjuvant therapy, which I oftentimes translate adjuvant therapy to patients by saying what adjuvant therapy means is you may or may not have cancer presently as we're having this conversation. We're going to talk about you receiving therapy that you may or may not need, but that we're essentially just too stupid to really be able to tell you whether you've actually got the problem or not. So let's imagine that these technologies that we're talking about, particularly early detection methods, allow us to be able to identify the problem, at a point we historically could not. as someone who's, lived through the journey, but also has, I know, had many interactions with patients who are approaching a cancer diagnosis, what do you see as the sort of pain points or the bottleneck in terms of actually trying to move like therapeutic investigation, not just use of currently available therapies, but investigational therapies up to this sort of, pre-radiographic detection moment or residual disease in blood by circulating tumor DNA or otherwise?
Okay. I have a plane tomorrow night, but I think I can get to that. First, behavioral economics tells us that the best technologies won't do anything if you expect people to go out and ask for them. People think going to ask to be screened is like turning yourself in to the police. So we have to make detection technologies passive. We need to make them pre-tumor, which is why CTC has issues. And we need them to be actionable beyond the diagnosis. So as an example, I'm an advisor to 2 different companies, one that only analyzes medical records. And in a study at Mayo, they found 20% of cancers in their sample test group 5 years earlier than Mayo actually found them, strictly based on analysis of their medical record changes in 20 different cancers. That's number one. Number two is that if you have the ability, as another company I'm working with does, to measure a certain kind of stem cell, called a V cell, very small, embryonic-like stem cell, those stem cells, when they're injured, make cancer stem cells. Now that's a big deal. And if we can detect through a simple blood test that you have cancer stem cells that are making this cancer, this subtype, and it's stage 1, now we're on to something. But it gets better because that kind of test can eventually identify the cancer pathway and within weeks test whether the therapy you're on is hitting that pathway without another biopsy. And in fact, our goal should be, how do we get rid of biopsies? Then the issue is, if we have that ability to see that, and we have the ability to tell you if a therapy is working, we still have the problem of distribution and affordability, which is what I worry about most with the new CAR-T generation of therapies, is they're unaffordable for nearly everybody without insurance and even with insurance. So if we can make testing as passive as possible, if we can show that not only can it diagnose you, but it can tell you if your therapy is working, and then make it cheap enough, and the tests that I'm talking about are around $100 out of pocket, not $1,000. If we can do that, then we have only to deal with the last problem, which is doctors who don't really care. Because they say, so you told me my patient has earlier pancreatic cancer, what do you want me to do about it? And that's a level of cynicism I'm not willing to surrender to yet. And I do think that if we know that we can tell if the therapy is working on intransigent cancers, and what I call the occult cancers, liver, brain, ovarian, pancreatic, if we can see those cancers through these stem cell analyses, now we can give people hope that we can actually do something about the diagnosis. 50 years ago, if you told me I had CLL, the next thing you said was goodbye. This time, there are like 20 different therapies I could be on. So there is hope, but how did I know I had leukemia? Did I go ask to get diagnosed? No, I did a physical, just a physical. I had no signs, no symptoms, but I had 180,000 white cells. So why did I do the physical? My doctor made me. He said, I haven't seen you in 2 years, come see me. If he hadn't done that, I would have found out the hard way.
Right. Wanted to kind of take a step back here, Shannon, to something you mentioned earlier. But we've talked a lot about designing a cancer, at least a treatment approach based on what we see from initial detection output. But can you talk a little bit about how we can use these same kind of approaches when we're understanding treatment-related toxicities, and in understanding how the body itself is responding and how that can kind of dovetail into maybe, an improved regimen or something along those lines?
Thank you for that. I think that one of the things that we're interested in is when we think about immune checkpoint inhibitors as well as CAR T, you're activating the immune response, but you can also sometimes overactivate it to really negative and sometimes deathly consequences. Unfortunately, they're rare, but it's something that we want to be able to monitor. And so we actually took a cue from the work that we were doing in COVID, where we were looking to see systemic immune responses and trying to understand through blood, saliva, and stool what systems are getting overactivated. And we think that this can be very beneficial for our cancer patients when we're putting on these novel therapies to see who might be more primed to have this overactivation, so that we don't necessarily completely stop the treatment, but look to see where is actually getting overactivated and can we treat that specifically so that they can still get the benefit from that particular treatment.
Great. And maybe a follow on to Gadi, I mean, you brought up a very interesting question about looking at the genome and all of its ancillary pieces in terms of, maybe trying to figure out and design a treatment approach. But what are we missing? And do you think this becomes kind of a chicken or egg problem, as in the more we learn from our sequencing efforts, the more we'll be able to kind of refine our approaches kind of moving forward?
Thank you for the question. I think we're missing one thing that we could do with more sequencing, wider sequencing and deeper sequencing, is figure out, for example, mutational signatures. Which mutational signatures that we see are relics of defects of DNA repair in the cancer that we could see from a blood test. And we could figure out if the cancer is has microsatellite instability, or has apobec activity, or other things like that are related to treatment or resistance, or homologous repair recombination deficiency, or HRD. We could get that from blood tests, from cell-free DNA. And even we don't need very deep sequencing in order to get that. So this is more signatures. We could also look for translocations and rearrangements that are not seen in typical sequencing of a panel of genes, and that's another thing that we're missing. But as I said earlier, one of the most important things is the dynamics. We could monitor not only whether there's cancer, what are the subclones of the cancer, using many mutations to monitor the cancer. Looking at a single mutation is not enough, because you cannot sequence deep enough to get the resolution that you need, because there are not enough molecules in the tube of blood. But if you look at many mutations throughout the genome, then you could get to a resolution of 10 to the minus 6 of molecules, and then figuring out if you have a recurrence of a disease, or a particular subclone is growing or dying, or things like that. So I think multiple time points measurements would allow us dynamics and also detection of all these events that we're not used to looking for. And another point I want to make is sharing of the data with research. So you're right that there's a chicken and egg situation. We need the data in research in order to figure out what's important and what's not. We don't have that data currently in research, sufficient amount, and most data actually lives in diagnostic companies, and patients should be able to contribute their data to research in order for us to actually find recurrent events or patterns of events that would tell us about whether the cancer would recur or not, and what's the aggressiveness of the cancer, et cetera. And that's something that I think is very important for patients to be able to contribute their data for research.
Got it. And then maybe one more for me, and let me plug the iPad again. So if you have a question, do let us know. But Jonathan, I wanted to follow up with something Greg had mentioned about having cost-effective approaches. And when you think about the underlying chemistry, what are the limitations here in terms of what we can do at a reasonable price point for your average citizen in order to make these detection approaches kind of more universal and accessible?
Yeah, great question. I think where we're at right now is the early stage investment in the chemistry. We're building the machines that can do these kinds of pioneering or initial capability studies. But down the road, it becomes a question for Russ, and I was going to ask him before if I could grab the mic. What kind of a yield do we need on the living tissue biopsy in parallel to the metaphor or the analogy of the blood culture, the microbiological culture? Those aren't 100% successful in identifying the pathogen. But as this sort of work proceeds, my answer to your question about the economics would be, when we get there with methods for seeing living samples for patient-specific tumor biology or subclonal tumor biology, we'll have put ourselves in a place where we could use cheap and affordable small molecules. And the actual science of deploying chemical on-off switches that we and others have built to see multiple biological targets in real time, that's not inherently expensive, but getting to the point where we have the clinical samples and the throughput and the pathways to say, how do we see our patients' CLL cells or our patients' tumor cells with high efficiency? So I'm curious, Russ, what do you think? What's the number? Is it 80%, 90%? The likelihood that a given biopsy becomes a Jenkins lab sample.
This is where our own success sometimes becomes a problem. And so we would usually experience somewhere between 80 and 90% success rate with a tumor specimen. One of the advances in the cancer that I treat, melanoma, is that we've been using some of our immunotherapies in earlier and earlier stages. And some of the specimens we get now are patients who only receive 1 or 2 doses, given every couple weeks, 1 or 2 doses. And we get the tumor specimen and there's no tumor left. And so that's technically a failure on our end. But it's the kind of failure we're totally fine with, because that means for that patient, there's actually a really good chance that after that leftover is taken out, again, that's the part that we get, they may not even need any more treatment. We don't have to have the conversation that, like Keith mentioned, where you may or may not have a problem. We're now getting smarter and smarter at saying, if we've actually done this limited experiment and it's worked brilliantly, you may be done. And again, that's early stage. This is all kind of evolving in real time now. But as we kind of read the winds of what's going on around us, we've noticed that we're getting 2 types of samples now: patients that got a little bit of immunotherapy and it's dead and we're happy, or patients where they've already failed the frontline immunotherapies, or the immunotherapies have failed them, and it's more and more aggressive and we don't have good ways of trying to kill that. So for those patients that are the patient population that I care about most, I see clinically, there's not a limitation in terms of the amount of tumor. Those cancers are growing. And so it's less of a problem, I think, for those stage patients. But it's something for those earlier stage settings where I think we're going to have to see how the field moves as well. In terms of what we need and how much, actually quality matters more than quantity. We can sometimes get very small biopsies, and if it's alive and growing, we can test a variety of different things relatively quickly. And we're starting to move now into blood cancers and cancers of the bone marrow as well, and so that's kind of an active area of investigation. But the hope is ultimately you could take what is now something that involves a bunch of technicians and principal investigators having coffee together and brainstorming to something that becomes fully automated, and everything soup to nuts is done by a machine, and everything is read out, and we have good correlatives to understand what it all means at the end of the day.
So, one thing that these technologies you're describing point towards is the idea that we'd be able to maybe start accelerating our work towards fully rational combination therapy regimens. My career started 25 years ago in oncology, and I used that phrase, rational combination therapy, at the dawn of my career, thinking that the genomics alone were going to get us flying along that curve. We now, 25 years later, have literally 3 approved sort of targeted therapy combinations, including immunotherapy and targeted therapy, but in chemotherapeutic regimens of the previous century, as you all well know, scaled up to higher order combinations empirically. But I'd like at least a couple of you to take a crack at what constitutes the path to rational combination therapy as you see it. So Gadi, maybe you take a first crack at that.
Sure. Thank you. I think that what we want to do is identify therapies that have non-overlapping mechanism of resistance. Because like we're successful in HIV, you want to essentially take all the cells of the cancer and give them drugs that even if they randomly became resistant to one of them, they cannot be resistant to all of them. So for that, we need to understand the mechanism of resistance. And now, based on what you said, how can we know for a patient which ones to give? That's why I think actually this spatial transcriptomics will help a lot, where you could take a biopsy and use, get both RNA expression and the images of the cancer and figure out what are the cancer cells expressed, there's also spatial DNA sequencing, so what are the mutations in the cancer, but also where are the immune cells, is there a barrier between the immune cells and the cancer and things like that. But in general, I think a combination of targeted therapy based on the drivers in the cancer, immunotherapy is based on the state of the immune cells around the cancer, and ADCs that are special to the cell surface markers of the cancer that actually carry with them the old chemotherapies with them into the cancer cells. I think these likely would work because each one of them has very different mechanism of action and therefore likely different resistance mechanisms. So that's my belief. Kind of.
All right, Russ, swinging it down to you then. Yeah, what's your version of rational combination therapy?
So I would have said the exact same thing 10 years ago. And I think the experience in melanoma in terms of rational combinations has been one where we take the best of what's around as quickly as possible and try to smash things together and see what works. And it has not been a successful strategy. And I think part of it is because we're still learning how immunotherapy works. And so implying that we actually have enough of an understanding to know exactly how resistance and response works, I think it leads us to a place where we're falsely assuming that we know what those combinations are. And I think that's the biggest challenge. And the best example, at least in melanoma, is the combination of targeted therapy with immunotherapy that was tried 3 times and technically is approved based on one of those trials, but no one really uses it. Because it doesn't look all that much better than immunotherapy alone, and the rate of side effects is quite high. So it should make sense that it works that way, but ultimately, and I don't want to sound like a nihilist here, it matters what ultimately works on the back end. And as a consequence, we try to inform what we do and what we study based on unbiased approaches, but then we try to vet it using these functional models to see, does the cancer even care about our ideas? Because at the end of the day, it doesn't read our textbooks, it doesn't read the papers that we write, it's going to fight back with whatever it's got, and we need to have some way of figuring out what that is. And so I don't know what the perfect approach will be, but I think it's going to increasingly require that we're studying the cancers that patients are walking in the door with, and not necessarily the cancers that are the easiest for us to study, or the model systems that are the easiest to handle.
All right, so both those comments, Gaddy's and Russ's, have to do with kind of the therapeutic strategy selection. So you make a selection, and you think you're on to something in terms of rational combination, but Shannon, how do we monitor better than conventional radiographic methods, for example, for judging response, non-response, which we know so many examples of how that does not give us great crystal ball insight for how patients are going to do long term. How do the advances we're talking about today relate to being able to understand quickly whether we're on the wrong track, right track, make course corrections?
Or toxicologically, too.
Right.
Well, I think it's really important to make sure that we're doing it better. I also think keeping in mind, too, the patient as well in terms of what they can access. For some of the patients coming into the city and having multiple blood samples and draws are just not efficient ways or ways that they can do when they're also not feeling at their best. So I think thinking through and also opening up to potentially biofluids that we haven't looked at before, whereas like in colon cancer, I'd love to look more at stool or urine and trying to understand what is acceptable for patients, even though stool sampling I've experienced is not something that patients are really excited about doing. Saliva, I think, is an untapped market. But even when we started working with saliva, a lot of our collection devices that we use actually destroy some of the biomarkers. But these are the ones that are FDA approved, so everyone's basing their knowledge of what's there. So I think for me, it's understanding how we're collecting these samples, what the patients are interested in using, and then really trying to take an open look at what could we find if we move away from some of these standard collection modalities and aligning it as well with other treatment time points. So I think for us, every time a patient comes into the clinic and has any type of sample, we look to engage at that time point so that our data is also related to other treatment that they're coming in for versus asking them to come in for a separate blood draw or something going forward. So I think if we do that, we can then start building all of that information that can help us. Right now, we're collecting in our glioblastoma patients about 20 samples a year based on their time course, which is too much, but we're hoping that by doing that type of sampling, we can identify what are those right time points that really matter for looking at these particular patients.
So, Greg, I take this question whatever direction you like, but it strikes me that much of what we're talking about is sort of diagnostic innovation, and it strikes me that a problem that we face in our entire ecosystem is that therapeutics are so highly valued. I mean, literally, like in the marketplace,
yes.
Diagnostics massively undervalued.
Yes.
But I guess what I want you to kind of opine on for us is maybe not so much just how do we fix that, but more like what do you see as the rate limiting step ultimately in terms of sort of translating these concepts to real clinical care?
Yeah, you read my mind actually, because when I hear this phenomenal work going on, it's new wine in a broken bottle, not just an old, but it's a broken bottle. So the way we do clinical trials, the way we finance healthcare, the lack of a derivative market for the healthcare sector, which is the largest sector of the economy, drives this crazy mutual assured destruction of drug prices going ever higher and payers getting more stingy about who can get what. So I have a different innovation I'm focused on to help not just these brilliant people, but patients in general. And that innovation question is, if something happened where we could make no more progress in biology of the sort we're all working on here, what would we do to lower the cancer rate in half? We're such techno-optimists that, like the GLP-1 drugs, we keep thinking there's going to be a shot that'll take care of cancer. But the fact of the matter is it's a system problem, and the system is the way we eat, the way where we live, the environmental hazards, the lack of access to health care in rural America. My hometown in Arkansas, 20,000 people, still doesn't have a resident cancer doctor. My dad was diagnosed in 2001 because he went to the doctor on a Tuesday. They only are available Tuesday and Thursday because that's when they came over from Memphis. If he'd gone on a Wednesday, he wouldn't have had cancer. So the innovation I'm focused on is, if we weren't making any more technical progress, what would we do to the system to start saving lives? And we know there's been a lot of progress in various things, and clinical trials in particular, but we're still so far away from matching this sophistication with the system that touches people's lives every day. And if we don't fix that, I'm afraid the breakthroughs here will only be available to well-off people in big cities. And that's my nightmare.
We had an interesting question from the audience here, but we've been talking a lot about treatment. What if we switch to the area of drug development? If you think about it, at this point in time, there's a single-digit success rate. Curious for each of the panelists, your thoughts on kind of what technologies you're dealing with could change that and help facilitate that. Maybe Gadi, start with you. And
Sure. We are now looking at cell surface markers that are unique to the cancer cells that could be used for either CAR-Ts or antibody drug conjugates to actually bring the drug to the cancer cells and only to the cancer cells. And I think I'm very excited about this ADC field, this antibody drug conjugates and CAR-Ts, and these bispecific molecules, because I think if we knew to bring the right drug to the cancer cells, we know how to kill cells. They're very potent drugs. It's just that currently they're given systemically, and therefore there's a toxicity issue. But if we could bring them only to the cancer cells or to the vicinity, then the toxicity level drops dramatically, and then we could increase the local concentration of the drugs. So to my mind, this direction is a very promising direction to development of new therapies. So that's one thing. But I would turn maybe to Russ to say, how can we test it on living tumors? I think maybe that's the next step.
And that's part of what we're interested in doing. And of course, we have the targets that we're in love with, that we think are the coolest. But I think one question more broadly that can be asked of any of the technologies that do this sort of living tumor biopsy is, If we could vet every emerging drug, whether it's cell therapy, targeted therapy, regimen, against patient tumor specimens, and find the failures early, would people listen? Would they actually, could you go to the people that are making the drugs that have already invested a lot in them and say, this is not going to go well, it's going to fail in phase 2 or phase 3, and can you save that money and say, now is time to pivot and reprioritize your portfolio or your strategy? Thus far, not many people are interested in hearing that message. As you can understand, especially with a relatively new and unproven technology. And without providing the particulars, I'd say that there's some companies that are more interested in others in testing things out to make sure that what they've got has a chance of success. But I think that's the way you go from single digit, or certainly 10% or less of phase 1 studies moving beyond to an improved rate, is you have to reduce your failure rate. There's good drugs out there, but it's saturated now with strategies that ultimately aren't going to work. Or they're not biomarker adapted. So they could work in a subset of patients, but we're not yet smart enough to figure out how to tailor the right drug to the right patient at the right time.
Can I throw one last concept in?
Please.
I've been focused a lot lately on N of 1 trials and N of 1 drug development. So Cancer Commons, many of you are familiar with, I'm on the board of, and I've been absolutely fascinated how many people you can help with an N of 1 study. Our patent system and our finance system push us away from drugs that are out of patent. Crazy. There's a company I've started talking to in Europe that does studies of people who have survived the worst cancers to reverse engineer and see what makes them the Berlin patient, so to speak. That's very exciting to me because blockbuster era is done. Even the GLP-1s will have a lot of variation for different populations. But the N of 1 studies will teach us more about us. And that, in turn, will help us figure out how to do better drugs in the future. Because when you're trying to see if drug X works for 100 million people, you're looking at the drug. You're not really looking at the people. If you're looking at one person, what works in that one person and why? And how did they survive this cancer? Then we're going to start learning things about our biology that will help all drug development, I think.
I'm with you. I mean, building from N of 1 to N of 10, and then take it from there. I mean, in other words, is it reproducible? Can you, do you have a pairing hypothesis that actually can predictably work in some number? I don't mean 10 for 10.
Yeah.
Building the evidence base in that way, that would be the flipping of the of our process. Because right now, companies, biotech companies anyway, are capitalized to get through phase 1/2. Proof of concept, come hell or high water. and by the time you're far down that track, you're right, the pivoting opportunity is kind of long gone. But building evidence base, in smaller units, if you will, as Greg is suggesting, I agree, is certainly offers much more sort of efficiency and, as Russ says, opportunity to pivot. I think we have to let these folks stretch their legs, find another session, and we have to hand over the mics to the next set of moderators.
I think so. I wanted to thank everyone for joining us. The timer here says we're 2 minutes over, although...
Not true?
I thought we were going to 5 till. Does anyone have a schedule handy?
I thought it was 11:45.
We can keep going.
Okay, fantastic.
We're going to keep going.
Awesome. All right, well, those who need to go on, they can. So Jason, you want to... sorry, Shannon was going to add to this.
Since my background's a little different, I'm a mechanical engineer, and when we're thinking about drug development and thinking about these tumors, one of the things we look at is the spatial organization of cells, not just who their neighbors are and what messages are, but how those forces of alignment also influence both the penetration of our cancer therapeutics into the drug, but also what those cells are doing. And so we see cell messaging and what cells are releasing in the tumor varies based on what the actual mechanical microenvironment is. And so I think that's often an underlooked area when we're thinking about new development for drugs, about how all of those other forces impact things.
Maybe could you just say a little bit more? I think, a lot of people in the field, the cancer research field, still don't quite understand what extracellular vesicles are, where they come from, which cells shed them and why, and what kind of information is in them.
Sure, absolutely. And so all cells in your human body, so it's a beautiful system because it allows us to monitor different cell populations, but cancer cells are known to release slightly more and also a bit more heterogeneous. So you can have really large ones as well as small ones, but large is still in the nanoscale. But inside of these vesicles, you can find DNA, you can find proteins, cytokines, and RNA. And what we found in pancreatic cancer when we take these patient-derived organoids, that when they're in a stiffer environment, they actually increase significantly more of these extracellular vesicles, and also they have different cargo that they're sending to neighboring cells. It's a form of cell-cell communication. So if you think about how these forces are both impacting the biogenesis of this message, but also what's packaged inside of it, it's something we should think about in terms of being able to both control and target these cancers therapeutically.
Very